Okay, let's start today's lecture. So today we're going to start a whole new pathway to study the so-called receptor tyrosine kinase, which essentially mediate growth factor signaling. And growth factors, of course, are very important for the regular proliferation, survival, and maintenance of most of our cells. And disruption of these signaling pathways result in human cancer, one of the most severe human diseases around. So let me start with the discovery of growth factors. Last night when I was surfing the web, I came upon this article, which was published 2 weeks ago on April 2nd in Nature. For those of you who are interested in reading, I highly recommend this article. This lady is the mother of growth factors. She's going to be 100 years old next Wednesday. It's too bad that we cannot time our lecture with her birthday. So the article is called 100 Years of Rita. Okay, so Rita Levi Montassini, she is the Nobel laureate for her discovery of the first growth factors, together with Stanley Cohen, who discovered epidermal growth factor, and she discovered nerve growth factors. So her life story is actually quite interesting. So Rita was born into a Jewish family in Italy at the beginning of last century, as one of the four children. Her father was an engineer and her mother an artist. So you can imagine at the beginning of last century, her father firmly believed that a position of women is at home, and that women should not pursue any professional careers or educations because that might interfere with their positions as a mother and a wife. So Rita knew from very early on that she has to have a professional career and she needed professional education. So she fought it very hard with her father, and she was able to persuade her father to let her go to medical school. So she attended medical school, went through the Department of Anatomy and Surgery. But just before graduation, she fell in love with basic research and decided to pursue that. And in particular, her interest is within the area of neurodevelopment and how the chicken embryo send out nerve axons to reach to the chicken limbs to direct their action. So when she was pursuing her research career, the Second World War started, and Mussolini published the race law in Italy, which essentially expelled all Jewish professors from universities and research institutions. So Rita was thrown out as well. But she did not want to give up her research career, so what she did is that she set up a bedroom laboratory at her home. So she purchased, so her family is pretty well-to-do, so she could purchase a microscope, an incubator, and ride the bicycles every day to the surrounding chicken farms to purchase fertilized chicken eggs to pursue her research. So during that time, she made a lot of studies on how the chicken embryos would send out these the ganglia, the group of nerves would send out nerves when you have signals from the chicken limb. And it was observed that if you remove all the chick limbs during the early development, then the number and the amount of nerves that send out from the chick embryos diminish quite a lot. So that experiment suggests that those chicken limbs might have sent out a factor that promote the nerves, promote the growth, the proliferation of these nerves and the differentiation of these nerves. So that was the idea. Now, at the end of the World War II, she was invited to come to the United States to give a seminar and do a few experiments. So the plan was that she came to the U.S., stay for 6 months, do a couple of experiments, and go back to Italy. And that stay extended to 30 years. So she worked at University of Washington, again doing the chick embryo experiment, try to figure out what that particular factor is in promoting nerve growth. And later on, she was joined by another colleague named Stanley Cohen, who was a postdoc at the time as well. So both of them worked together, try to identify that particular factor that can induce nerve cells to proliferate and to differentiate. So they were trying to look for what are the other things that can respond or send out that factor to allow chick embryos to send out nerves. So one the thing they found is that if you take a tumor, a sarcoma from the mouse, and co-culture it with the chicken embryo, the chicken embryo will also send out mass amount of nerve mass. And so this is one of those results. This is a neural ganglia from the chick embryos without any co-culture factors. This is what it looks like. And if you co-culture with either a tumor mass or chick limbs, then you get this nerves sent out massively from the central nuclear ganglia. And I should note that this particular piece of figure is actually hand-drawn by Rita herself. She's actually an artist at the same time. So she actually personally hand-drawn all her figures in her early publications and designed all her own clothes. So that article had many nice pictures of her early days. So this is actually a very interesting older generation of scientists. So and then during the search that they were trying to purify this factor, so they need to find other sources of this factor. So they experimented with a lot of things. And what they found is that in addition to chick limbs, mouse sarcomas, there's another place where they found a huge abundance of this factor. Where else, of all places? Snake venom. So then everybody laughed at them. Now, how can you put what's in common between a chick limb, a tumor from mouse, and snake venom? So everybody said they're dreaming. How can you have a nerve growth factor that's present in all of these weird places? So she had to really defend herself and keep continuing. Finally, in 1971, after more than 20 years of work, she's able to purify that factor and sequence it and name it nerve growth factors. And at the same time, Stanley Cohen, working closely with her, but using another system to study growth factors, it found that nerve growth factor, NGF, is not the only one. There's another thing that he named epidermal growth factor, EGF. That's something that we hear all the time. And he was able to purify that factor. And because of this discovery, both of them were awarded the Nobel Prize. And Rita has been working with NGF ever since. And she still has a laboratory in Italy now, approaching 100 years old. Her students still talk to her sometimes about science. And her mind is still sharp. It's very interesting. So nowadays, we know that there are many growth factors. The ones that we're going to talk, so these are the ones that belong to, that use similar mechanism for signal transduction. So there's nerve growth factor, EGF. PDGF is platelet-derived growth factor, and fibroblast growth factor, insulin-like growth factor. Of course, you don't need to remember the details for each abbreviation, but you need to know what they are. So in our lecture, I'm going to mention EGF a lot or PDGF a lot. So just understand that these are growth factors that when you add it to the cells, that make the cell proliferate. So in the laboratory, when we do tissue cultures, when you grow mammalian cells, one of the major components you need to add is serum. And what are the things in the serum that's so important? These guys, okay? EGF, PDGF, insulin, these are all important growth factors that allows the cells to survive and to proliferate. So how do they allow cells to grow and to proliferate? So they function through growth factor receptors. And these are the different growth factor receptors. Again, you don't need to memorize each of them, but you notice that there are a lot of similarities among all these different growth factors, receptors. This is the EGF receptor, insulin receptors, NGF receptor, PDGF receptor. So one thing that they have in common is that these are normally monomeric molecules, that they go through the membrane once. There's three components. They have an extracellular domain that binds to ligand. They have a transmembrane domain that anchors the receptor on the surface of the cells. And they have a cytoplasmic domain that's responsible for transmitting the signals inside the cells. And among all of these cytoplasmic domains, they all contain the so-called tyrosine kinase domain. So this is a motif that when it's active, it functions as a kinase that put the phosphate group onto the tyrosine residues of the substrate. So it has a specific activity for tyrosine residues, not for serine threonine kinases. Those are the ones that we have been talking about. Those are serine threonine kinases. These are tyrosine kinases, okay? So some of them have essentially one big kinase domain, and some of them have two smaller kinase domains with a little linker region in between. We call it kinase insert region. But essentially, they have, what's in common is they have tyrosine kinase activities inside the cells, okay? So how was the tyrosine phosphorylation actually discovered? Before people studied tyrosine kinase, everything that all the kinases that we've been talking about, PKA, PKC, CAM kinase 2, they're all serine threonine kinases. So it was actually by surprise that the phosphotyrosine was discovered. One typical experiment was actually done by this guy, who's actually quite famous, Tony Hunter. He is still at the Salk Institute, where he actually performed this original experiment. So he was also the one who wrote the review article many years ago about 1001 kinases. So his specialty is tyrosine kinase. So about 25, 30 years ago, when Tony was still a postdoc at Salk Institute, he was doing this laborious and tedious experiment, trying to analyze the content of that phosphoprotein, whether it's phosphoserine, phosphothreonine, or phosphotyrosine. It's just a routine work. So people know that when you add growth factor to the cells, they become phosphorylated, but they don't know what residue was phosphorylated on. And it was assumed it's serine and threonine. So he needs to confirm that. So the way he does the experiment is that you label the cells first with orthophosphate, but it's P32 labeled so that we can trace the radioactivity. So this is, we're talking about 5 millicuries to 10 millicuries per experiment. It's really very screaming hot. So you add this to the media, and you wait for it to get absorbed by the cells, and then all the ATP pools in the cells are saturated with orthophosphate. And so all the newly synthesized ATP, nucleic acid, everything will become labeled with the P32. So then after that, so the labeling goes for overnight or 16 hours. Then after that, you take up these very screaming hot cells, and then you lyse them. And then you purify the P32 labeled protein by antibody affinity column. So if you're interested in the protein, for example, EGF receptors, you use the antibody against EGF receptors to isolate that. And if it's phosphorylated, it will have P32 label on it. So then, but there are a lot of P32 labeled proteins in the cells, and if you use the antibody column, it still brings out non-specific signals. So you need to further purify that or resolve that on the SDS gel. So by running the whole thing on the gel, you separate the protein based on their size, okay? And then you have an individual band that's highly purified, so he cut the band out and grind it up and elute it and try to elute the gel. So this step, everything, one sentence takes a day, okay? And then after eluting the protein, the elution efficiency, of course, is not very high. So then he get the pure protein, and then you digest with 6 normal HCl. What does 6 normal HCl do to peptide bond? It breaks it, right? It hydrolyzes it. So you break it into individual amino acid. And so in that mixture, you have unlabeled regular amino acid, all 20 of them, and then you have the phospho labeled. If it's serine, it's labeled as phosphoserine. If it's tyrosine, it's phosphotyrosine. Then you need to resolve these 3. And of course, they're too small to be running on a regular gel, so you need to run on a thin layer, sort of a chromatography plate, but run a 2-dimensional electrophoresis. In one dimension, you run at a pH 1.5 solution, and the other dimension, you run at a different pH, 3.5 solution. So then you try to resolve all 3. And then you put that plate on the gel, you do autoradiography. So you start with about 10 millicuries of P32, and end up here you get about 100 DPM. And many times you need to expose for a week before you can see the signal. And this whole thing takes almost a week. It's a very long process. So it was a legendary story. So the first time Tony did it, by the time he get to this stage, it's already a Friday afternoon. Okay, it's very hard. You have to work with all these radioactivity, try to protect yourself, at the same time try to grind up the things, which tends to splash everywhere. and you need to clean up after you. So by the time he ran the final stage, ran the gel, it's very late. And after he ran the pH 1.5 dimension, he realized he ran out of pH 3.5 buffer, and that happens all the time. And of course, as a human, most of us want to be lazy on a Friday afternoon. So he went to a colleague of him and asked to borrow some pH 3.5 solution. So the guy said, yes, you're free to borrow this bottle, but the bottle, I need to warn you, has been sitting there for 2 months. So it's very old. So he said, well, what the heck. So he ran it, and then this is the gel he got, and it was just the dream gel. So this is what the chromatography plate looks like. He ran the first dimension at 1.9, ran the second dimension at 3.5. When you run it, you can purchase the standard phosphoserine, phosphotyrosine, phosphothreonine standard from Sigma. And you run it with them, and you can stand with anhydrine, and you know where they are. And this is the autoradiography, okay? So the dotted line indicates the position of the standard. And then when he does radiography, he found that most of the phosphorylation is on tyrosine residue. So this is what it looks like. And this is the first report of that. It was extremely exciting. So then what happens is that he needs to repeat the experiment. So he decided that when he repeated the experiment, it's such an important discovery, he wants to make sure every step is correct. He made all the buffer new, make sure everything's done correctly, and he repeated the experiment, the spot disappeared completely. He repeated again, it disappeared again, so it's called a beginner's luck. So then he tried to troubleshoot. So what did I do differently from the first time, from the second, third time? And he realized that the difference is in the last buffer that he borrowed from somebody. And they use exactly same recipe. There's not much to it. There's 3 components in that buffer. So the only difference is the first time he borrowed that buffer, the buffer's been sitting there for 2 months. And when he made the new ones, they're freshly made. So he decided to, okay, he's going to let the buffer age. So he made the buffer again, let it, every time he aged it for 2 months, he get this beautiful result. So when I was a graduate student at Salk, working next door to this guy, we all know that when you make the buffer, you have to make it in a slightly different way, because the original buffer is, if you made the original way, you have to let it age for 2 months. If you don't want to wait, you have to change the pH a little bit. But that's how it's discovered. So all of these scientific discovery happened with some sort of an accident in a way. But a good scientist is able to really observe and not give up, and really think through the troubleshooting step and be rewarded with the great discovery. So this is, so it's now established that all these growth factor receptors are tyrosine kinases, but how do they get activated? When we think about G protein coupled receptors, we know that ligand binding causes the conformational change of the receptors. And that's pretty easy to imagine, because there's 7 transmembrane domains. So a little conformation change here and there is easily exposed certain loops so that it can bind to the downstream molecules. But how much variations can you get with a single monomeric molecule with only 1 cytoplasmic domain? You can tweak the molecule this way, that way, you change the conformation, but by it alone, it's hard to imagine it will expose certain things that were never there. So one thing, it turns out that it's not just the conformation change of a monomer. The binding of the ligand actually brings the 2 monomers together in a process we call dimerization. They make them form a dimer. So in some cases, it's a monomeric receptor that once bind to ligand, 2 ligands together form a dimer. In other cases, the dimers actually are preformed, but they're not in the active form. And when you have ligand binding, that change the conformation in the transmembrane domain. So used to be dimer might be forming, preformed dimers might exist in such a conformation that the cytoplasmic domains are actually far away from each other. And when you have a ligand binding, the transmembrane domain switched so that now the 2 cytoplasmic domains can actually get together. So the consequence of that getting together is that the tyrosine kinase, the little residual activity of tyrosine kinase, is able to phosphorylate each other, okay? So we call this event autophosphorylation because it's the same type of molecules phosphorylating the same type of receptors. So they autophosphorylate, and then that additional phosphorylation events would then fully activate the kinase. So there are experiments that's done to show that this dimerization is really important. You can make point mutations in the receptors that destroy this ability to dimerize, but still retain the full kinase activity. And people shown that in that case, when you add the growth factors, they don't dimerize, they don't get activated. So dimerization is the first key step in the activation of these molecules. Okay, so the next step after dimerization is the autophosphorylation. Okay, so one of the cytoplasmic domains, they physically interact with the other one, and then they bind to ATP, and then they phosphorylate each other on multiple tyrosine residues. A tyrosine kinase many times, any proteins, if you just randomly look at the sequence, it probably contains many tyrosine residues. So for the tyrosine kinase, when it autophosphorylates each other, most of the times it's not just one side. They actually phosphorylate multiple tyrosine residues. So there are two types of, when we really strictly talking about autophosphorylation, this is just the observation of a whole group of receptors, like EGF receptor phosphorylate itself. But when you look into more details, you realize that there are actually two types of autophosphorylations. They're both called autophosphorylation, but they're not exactly the same. One is intermolecular autophosphorylation, meaning that there's one molecule of EGF receptor phosphorylate the other molecule of EGF receptor. So it's cross the molecule phosphorylation. We call it intermolecular autophosphorylation. And there's another type called intramolecular autophosphorylation, meaning that this kinase molecule autophosphorylate a site in the same molecule and not to the other one. Okay, so when we do experiment, in many cases, when you add EGF to the cells, you observe phosphorylation. It doesn't really tell you whether it's intermolecular autophosphorylation or it's intramolecular autophosphorylation. So I'm going to leave you with a question that, giving you all the molecular tools that we can do nowadays, you can do DNA mutations and all sorts of things. Can you think of an experiment to distinguish between the intermolecular autophosphorylation and intramolecular autophosphorylation? So it's not an easy experiment, so take some time to think through it, okay? And then it's not going to be in the exam, so don't worry. But just think about it, it's fun, all right? How do you distinguish between intermolecular autophosphorylation and intramolecular autophosphorylation? But so the final consequence is you get all these different phosphotyrosine residues. Now, what do? How does different phosphotyrosine residues send the signals downstream? It turns out that these phosphotyrosine residues can serve as docking site for binding to proteins that tends to like them. So there are proteins that have specific domains. We call them SH2 domains or PTB domains. So SH2 stands for Sark homology 2 domain. So Sark is the first tyrosine kinase, first oncogene that's ever been discovered. And it was discovered where? Anybody know? Here, at Cal. By who? Anybody know? Who's still working in the department? Not me. I'm too young for that. The chair of our department now, Steve Martin. Okay? So, who's that? Well, if you're EMCB, he has the final authority to sign all your diplomas and papers. Anyway, so he's the one who discovered Sark. Sark stands for sarcoma. It's a tumor that's isolated from the chicken. Okay? So Sark is the first oncogene ever discovered. And Sark is a tyrosine kinase, but it's not a receptor tyrosine kinase. It's a cytoplasmic tyrosine kinase. It has many domains. So a lot of proteins contain this domain that looks very similar to a region in Sark. That's why we call it Sark homology domain. So there's Sark homology 1 domain, Sark homology 2, and Sark homology 3. The SH1 domain is the tyrosine kinase domain. All tyrosine kinase look similar. Okay? SH2 domain is what we're going to talk now. Okay? And PTB stands for phosphotyrosine binding domain. So these are modular, independently folding domains. They're about 100 amino acids in length, so it's relatively small, and they usually exist in a certain portion of a polypeptide chain. In the three-dimensional folding, they can independently fold into a structure that binds to phosphotyrosine-containing residues. So if you have a protein that has an SH2 domain, it will recognize a molecule that has phosphotyrosine in it. So when you have a receptor that has many phosphotyrosine residues, each of those phosphotyrosine residues can function as a docking site to allow proteins that recognize that residue to bind. So there are actually many study variable SH2 domains or PTB domains. Each of them will recognize slightly different sequences around phosphotyrosine. So this is kind of a three-dimensional structure of an SH2 domain in a gray area. So this is a part probably connected to the rest of the proteins. This is an independently folded SH2 domain. And on top of it in the color are the phosphotyrosine-containing peptide that can bind to this domain. So this is the SH2 domain of SARC residue, and this is the peptide that SARC prefer to bind. Now, the first thing you want to look at is here, there's a pocket. And behind this gray veil pocket sit this tyrosine, phosphotyrosine group. So this is, tyrosine, remember the structure, is a pretty bulky group, okay? And once it's phosphorylated, this phosphotyrosine sits right inside of this pocket very comfortably, okay? So this is one pocket. The second pocket is right here. So this is another pocket where the residue, third residue downstream of phosphotyrosine, which happened in the case of SARC, happened to be isoleucine, sits in this particular pocket very well. And the rest of the residues between the two can sit pretty snugly on the surface of this SH2 domain. So the SH2 domain here is kind of like a two-pronged plug, fit into the socket, two socket here, and the rest of them sit on top, bind very nicely. Now, different SH2 domains, they all have the same pocket here. That's the phosphotyrosine. But then a protein can have many tyrosines, and the residue around the tyrosines, of course, are all different. So some of the pocket here, like in this case, fit isoleucine, but other pocket may fit other amino acid. And the surface structure, shape, all are different. So different SH2 domains will all recognize phosphotyrosine, but the residues around phosphotyrosine may be different. So each SH2 domain has its own set of phosphotyrosine-containing peptide that it prefers to bind. It all depends on how complementary the shape is. Okay, so there are many phosphotyrosines, phosphotyrosine and the surrounding peptide regions, and there are many SH2 domains. As long as the two fit very well, then they will bind. Okay, so for SH2 domains, they all have the phosphotyrosine, and then there's another pocket that's determined by the +3 position, meaning the residue's carboxy terminal to the tyrosine. Okay, so PTB domain is slightly different. So SH2 domain likes phosphotyrosine plus the 3 or 4 residues carboxy terminal to it. So that's where the specificity comes in. And PTB domains likes phosphotyrosine plus residues amino terminals to it. So these two phosphotyrosine-containing domains look at phosphotyrosine in a slightly different context. Some proteins have SH2 domains, some proteins have PTB domains, others have both of them. So they can bind to different phosphotyrosine residues. Now, when you have a receptor, as we said, the receptors will autophosphorylate to produce many of these docking sites, and each docking site is able to bind to a protein that contains an SH2 domain that fit this particular residue and the context. So if you have 4 phosphotyrosine residues, you can bind to 4 different molecules. And that's how signal gets sent out. So you add one growth factor, you immediately activate multiple different downstream signals. Okay, so what are the downstream signaling molecules that's activated by growth factors? So independent of the study on phosphotyrosine kinase, receptor tyrosine kinases, people always been studying human cancer, and they discovered another molecule that's very important for cancer, so it's called RAS. RAS was discovered by Bob Weinberg at MIT, who's still active. So as I said, SARC was the very first oncogenes ever discovered, and it was discovered from chicken, so it's a chicken avian oncogene. RAS was the first discovered human oncogene, okay? And of course, now we know SARC and RAS exist in all the cells. Doesn't really matter where they come from. RAS is, we've heard about RAS before, it's a small GTPase. And as the trimeric G proteins, it switches between the two forms, the inactive GDP bound form and the active GTP bound form. In the case of trimeric G protein, the G alpha subunit, it's a large protein. So it has intrinsic GTPase activity. And whenever it binds to GTP, it hydrolyzes it. Now, in the case of RAS, this is a very small molecule. It's not one of the gigantic transmembrane proteins. It only has 170 amino acids. It's very small. And all of its amino acid residues is devoted to one function, a GTP binding and a small GTPase. But this GTPase activity is very, very weak. Okay? If you leave itself, let it to hydrolyze GTP, it takes minutes. That's a long time. So it needs the help of an exogenous gap protein, GTPase exchange factor, in order for this switch to happen. For the trimeric G protein, G alpha subunit also contains part of the gap activity. So that hydrolyze speed is much faster. But for RAS, you absolutely need an outside gap in order for this to happen. And similarly, you absolutely need a guanine nucleotide exchange factor in order for it to go from GDP to GTP state. Now, when I mention this one, I want to correct one mistake in previous handout that I copied from the last edition of the textbook, which turned out to be wrong. Okay, so in the previous graph that you had in the G protein signaling, I have another molecule here, it said GDIs. That's put at the wrong place. GDI should be on this side, not on the other side. So please correct that. One of your students actually came to my office hour last Friday and point out the mistake. So I want to thank him for that. So here, again, so so we know that RAS is a small GTPase. And what are the evidence that RAS is actually involved in receptor tyrosine kinase signaling? There's several evidences. First, we know that when RAS is activated, it needs to bind to GTP. So people have been studying, because it's a GTPase, they said, okay, under what condition would RAS start to bind to GTP, meaning it's activated? And they were trying all sorts of things. It's like chicken sink experiment. And in the end, what they found is that growth factors. When you add EGF to the fibroblast, you notice that RAS bind to GTP. So that would indicate that the growth factors could possibly activate RAS signaling. So with that, then can you go another step beyond to say that RAS is important for growth factor signaling? Not necessarily. When you add a growth factor in the cells, millions of things happen. Some things can be important, something can be a side effect. Sort of it's a correlation, but it's not really causal. So they decided to do a further experiment. If RAS is really important for growth factor signaling, then if you microinject cells with an anti-RAS antibody to block RAS function, then you would expect the cells will no longer proliferate in response to growth factors. And that's exactly what happens. So they take the fibroblasts, they microinject the anti-RAS antibodies into the cells, then they treat the cells with two growth factors, PDGF and EGF, and then they look at the cell proliferation, and these cells never proliferate. So that suggests that RAS is necessary for growth factor signaling. If you block it, you don't have growth factor signaling. But the next question is it sufficient? Yes, it's important, it's necessary, but is it enough to have it? So the way to do that experiment is that what growth factor does is to activate RAS. Now, what if you find a mutation in RAS that keep it in the GTP-bound state, a constitutively active RAS? Then in this case, if it's sufficient, then you wouldn't need growth factors because the cell would recognize this as it's being activated by growth factors. So they did the same experiment. There's a natural occurring mutation, RAS-D, in human cancer that keep it in the GTP-bound state. When this molecule is introduced into the cells, even without growth factors, the cell just keep growing forever, like a cancer cell. So that suggests that the RAS is not only necessary, but also sufficient. All you need is active RAS. You don't need any signals upstream of RAS. Okay, So RAS is very, very important for signal transduction. Okay, so now we have a partially linear pathway. We have EGF bind to EGF receptors, allow its dimerization, autophosphorylation, and activation. But how does EGF receptor activation get to RAS activation? We know RAS is a small GTPase. In order for it to be activated to go from GDP bound state to GTP bound state, you need a GEF. You need a guanine nucleotide exchange factor. In order for this guy to transmit signal downstream, you need a SH2 domain containing protein. Okay, so we need two things to mediate this activation. We know that RAS is so small, it does not contain an SH2 domain. So RAS cannot bind to EGF directly. It needs to be something else. And as I said, RAS needs activation of GEF. So whatever the signal intermediate has to have at least fulfill both conditions. First, that intermediate has to have either SH2 domain or a PTB domain in order to be able to bind to the receptor. And second, that whatever that intermediate is, it has to include a GEF activity in order to be able to activate RAS. Okay? So people have been searching for the missing intermediate for many years. And I'm going to tell you another experiment that's done here at Cal, just to show you how great our department was. Sorry. So Jerry Rubin's lab, who was, he's now at Howard Hughes. When he was in the department in the early 80s, in actually mid-80s, he was studying Drosophila eye development. So Drosophila fruit fly has a very interesting eye structure. It's a compound eye. It forms each eye of Drosophila. This is one big eye of Drosophila, tiny. It's formed of 800 little individual eyes, and each of them is called ommatidia. Okay, and each ommatidia has exactly 22 cells, and out of that 22 cells, 8 of them are neurons, photoreceptor neurons. So that's the structure shown here. So these are the photoreceptors, and they send axons to the brain. Now the photoreceptors, these are receptor molecules. There are 8 of them. Each one has 8 of them, and it's called from R1 to R8. Okay, and they form the structure that looks like this. If you do a cross section of these photoreceptors, you can look under the microscope, and you can see the similar pattern, identical pattern for each eye, with R1, 2, 3, 4, 5, and R7 is on the corner. Now, there's a naturally occurring mutant. That mutant, Drosophila, does not have R7 right here, this one. Okay, so if you look under the microscope, you see that there's R7 is missing. Now, these different photoreceptors each are responsible for taking a different spectrum of light. So R7 turns out to be responsible for sensing UV, ultraviolet light. If you take away any of the other R's, neurons, so R1 to 6, the Drosophila essentially died. Because think about it, if you're blind, how can you fly around and find food? So they can't do it, so they die. The only exception is R7 and R8. When R7 is missing, they don't see UV, but they're okay under normal light conditions. So these flies actually survive. And so using this, so they call this mutant fly that doesn't have R7, 7-less, okay, because it doesn't have 7. So they look, so what are the other mutations beyond that's not on the R7 that also, when it's mutant, also cause the 7-less phenotype? And with that, they found the R8. Okay, the R8 turns out express a ligand for the 7-less. And so people who work with Drosophila have extraordinary imaginations. So they named that ligand molecule bride of 7-less, because this is the receptor that takes the ligand. So it's called the bride of 7-less and abbreviated as BOS. And the bride is always the boss, of course. So now you have a ligand that doesn't help with anything. Ligand bind to a receptor tyrosine kinase receptor, receptor tyrosine kinase. So that's your traditional pathway. What are the next steps? So in order to find the next step, then they design a very nice genetic screen. Drosophila is a great genetic tool. So what they did is they make a temperature-sensitive 7-less. So at the non-permissive temperature, at the permissive temperature, everything lives just fine. You have fully expressed 7-less. But at non-permissive temperature, the 7-less gene is disrupted, so you don't have any R7. At the intermediate temperature, when half the 7-less gene is disrupted, you get half the amount of 7-less protein. Okay, so some of the cells have R7, some of the cells don't. But overall, the Drosophila lived pretty nicely. So they argue, now this is the threshold. So the R7 amount is right above the threshold level that's important for R7 to form. Now, at this moment, if you give another hit, if you mutate another molecule in the system, then you'll probably just destroy the whole thing. So they did that intermediate temperature, they add another mutation, and they look for what other mutations can make R7 disappear. So they found 2 molecules. One is SOS, stands for son of 7-less, because it's downstream of 7-less. So of course, you have a bride, you have a 7-less, you have a son. And the GRAB2 is not named because this GRAB2 turns out to be discovered in the mammalian system first. So it's much less imaginative. So they just use the same name in Drosophila. Now, so there are 2 of them. Okay, so where do we place them? It turns out that GRAB2 is a very small molecule. It has 2 types of stuff. It has the SH2 domain, which is what we're looking for. Okay, so it has the SH2 domain that can bind to phosphotyrosine, bind to the receptors. So that's great. But it doesn't have a GEF. Instead, it has 2 other domains called SH3 domain, the Sark homology 3 domain. Now, what are the SH3 domains? It turns out the SH3 domain is kind of like SH2 domain. It's, again, a protein interaction domain. It's about 100 residues or so, and it's also independently folded. But instead of recognizing phosphotyrosine, it recognizes what they call the proline-rich sequence. So this is the structure of a Sark SH3 domain. Again, the gray part is the Sark SH3 domain. And on top of it, you can see the pocket where one proline residue sits, this green, blue one, another pocket where this proline-rich sit, and then the rest of the residues spread out. So these proline residues sit on the SH3 domain, provide the structure motif, and then the rest of the residues spread out on the surface of the SH3 domain. So whenever you have an SH3 domain, it will recognize another protein that has multiple proline residues next to each other in close proximity, and also the specificity of that recognition, again, is achieved by the residue between the 2 proline residues and around it. So depending on how they fit the surface area of a given SH3 domain. So there are, again, many SH3 domains and many proline-rich sequences. So when GRAB2 has an SH2 domain, which allow it to go to phosphotyrosine, it also has 2 SH3 domains that allow it to bind to the next molecule that has protein-rich sequences. So it's essentially a bridge. GRAB2 is functioning like a bridge to bridge the receptors with another downstream molecule. That's all it does. So we call it an adapter molecule because essentially that is an adapter and nothing else. Okay, that's the thing we've already talked about. But GRAB2 does not have a guanine nucleotide exchange factor activity. So we still need to look for that. Now, it turns out that actually SOS has that. So SOS protein has a lot of prolines, and it's a proline-rich sequence, and also have the guanine nucleotide exchange factor activity. So it's actually a pretty nice fit. So GRAB2 would bind to phosphotyrosine residues in the EGF receptor, and then bridge the interaction with SOS, and this has a guanine nucleotide exchange factor activity, which leads to RAS activation. Okay, so I'll go through the whole process for you, walk through the process. So this is the unstimulated state. You have monomeric EGF receptors on the cell surface. Now RAS, small GTPase, I should mention it, it's also a membrane-bound protein. It's not completely sit inside, but at the beginning of the RAS, there's a little lipid motif that allow it to bind to the plasma membrane. So again, this is like an effector molecule. It sticks in the membrane, but it cannot be activated by the receptor directly. When you have EGF binding, it induces dimerization of EGF receptors, allow it to autophosphorylate itself to create a lot of phosphotyrosine residues. And then that allows the GRAB2, which is normally a cytoplasmic molecule, to come to the receptor to get near the plasma membrane through the SH2 domain phosphotyrosine interaction. Okay? And once GRAB2 binds, it initiates the assembly of a whole complex. So GRAB2 binds here, and that recruits SOS, which is also a cytoplasmic protein, to come over to bind to the SH3 domains in GRAB2 through protein-rich sequences. And then SOS can then bind to the RAS next door, allow the RAS to undergo a conformational change, which then kick out the GDP. Okay? And then because the cells, as I said, has a more GTP than GDP, you get GTP to bind to RAS. Okay? And then from there, RAS can send out downstream signaling activities. So this is the major step going from the ligand to RAS activation. And I should mention that it's not just RAS that's getting activated, because that only accounted for one phosphotyrosine residue. In the receptor, there are many phosphotyrosine residues. So you get a slurry of other activities that contains either SH2 domain or PTB domain to bind to these different phosphotyrosine residues. The examples are, for example, phospholipase C. Remember phospholipase C that we talked about last time? Phospholipase C generates IP3 and DAG, eventually leading to activation of protein kinase C and calcium. So in this case, there's a different phospholipase C isoform of phospholipase C gamma. It does the same thing, but phospholipase C gamma has SH2 domains, so it can bind to one of those phosphotyrosines. If it finds the right fit, it will bind. And then that will lead to increased calcium concentration and changes in metabolism. There are other molecules that we're going to talk about later on, PI3 kinase, phosphatidylinositol 3 kinase. That molecule is very critical for the survival of cells and for the growth in size of cells. So that molecule also contains the SH2 domain, and it can bind to yet another phosphotyrosine residues. But these molecules don't necessarily bind to every single one. It all depends on whether you have the right fit phosphotyrosine or not. And it turns out that most of the receptor tyrosine kinases that we talk about have binding site for all of these guys. So think about the variety of biological responses you will get when you add growth factor. You get RAS activation, which we will talk in the next lecture about. It's activating a RAS MAP kinase pathway, which directly stimulates cell cycle progression. And then you get phospholipase C activation, which will increase calcium and change cellular metabolism. And then you get increased survival signal to tell cells to survive, and then increase metabolism to grow in size. So all of these actions essentially coordinate with each other at a single point of growth factor receptors. So all of these activities all happen at the same time to allow cells to live, to proliferate, to increase metabolism, to grow in size, and eventually become, one cell becomes two. Okay? So we'll talk about this next time. These pathways, of course, are targeted human cancers.